Hello and welcome to this webinar about the European Variation Archive. In this webinar we'll talk about how we can access this archive at EMBL-EBI programmatically, and that will be mostly using our API. The presenters here are I am Gary Saunders, and beside me we have Pablo Arce Garcia. So I'll give a brief introduction to the European Variation Archive, and at EMBL-EBI we have a suite of resources that take us through the biological life cycle, from genes and genomes with things like Ensemble and the European Nucleotide Archive, through resources for genes, protein and metabolite expression, and then resources for protein sequences, right the way through to resources that cover the literature and ontology that govern the complete process. And in 2014 we launched the European Variation Archive, which comes in at the top of this tree, which is a specific resource at EMBL-EBI that deals only with genetic variation data. The EVA is a submission-based data sharing and analysis platform. Importantly, it's for all types of genetic variation: small nucleotide variants, multi-nucleotide variants, indels, and right through to structural variations. The samples that we analyze can be germline, somatic, within populations, or the variants that we have can be potentially even between species. And the point of the EVA is to share variation files and variant data from a single resource here at EMBL-EBI. So why do we want to archive variation data and variation files? The first reason is that we can administer file accessions. And these stable file accessions can be cited in publications, and they can be shared between researchers and labs. Because they're entirely stable, any data that's within that accession does not change. So as a postdoc moves on to a different lab, or as a project changes over time, anything that's accessioned will not change and can be traceable via the literature or via any mechanism that we use. The second reason is that we administer variant accessions. These are individual accessions for each variant within a VCF file, variant call format file that we use at EVA. And the variant accessions we use are the same as DB SNP. So we have an SS identifier for a submitted SNP, and periodically these are clustered into reference SNPs. At EVA, because we use the variant call format VCF file, any variation is represented as a change against a reference genome sequence. So if we look under the hood at EVA, this is our data model. We have the submitter that comes to EVA with an analysis object. That analysis object describes the samples that were analysed, the methodology or pipeline that was used to call the variants, and the genome against which the variants are placed. And it's these analysis objects that we host at the EVA. The EVA content, we have roughly 100 short variation studies, 40 of which are human. We have over 150 structural variation studies, 118 human. And within these studies, we have more than 1 billion variants that have been submitted from the community. This is a screenshot of our study browser at the EVA website. It has some filtering options on the left-hand side that allow you to filter these studies. And then when you access any of these studies, we have some more study-specific metadata, which describe the title, the number of samples that were analysed, and also give you access to the flat files via FTP. So that's the brief introduction I wanted to give. Now Pablo will talk more about the EVA API.
Well, we are going to take a quick look in the EVA API. All URLs in our API start with a fixed part that is common to all the endpoints, and then is followed by a variable path that in most of the endpoints follow this pattern. We can find a category followed by an ID or list of IDs, the resource that we want to query, and a set of filters or options. Here we can see a complete example of a working URL in our API that this can be used to get all the variants for a given gene. Currently, EBI just allows HTTPS traffic. So if you do an HTTP call to one of our API endpoints, it will be redirected automatically to HTTPS. And if you are using a web browser, this is done automatically and you won't notice anything. But if you are using a HTTP client in your programming language of choice, you can see that this is not automatic, so it's better to call directly to an HTTPS URL to avoid problems. Currently, we just allow queries, so you have to use the HTTP GET method when you are calling to our API, and we will return to HTTP codes. 200 if the URL is okay and the query is well formed. Even if there are no results, we will return an empty result list. We will see the format of the output later. And if the query is not well formed and the URL is not valid, you will get a bad request code. Well, we have 5 main categories: variants, segments, genes, and files and studies. We will see those categories now. Files and studies are mainly used for metadata of the studies and files that we archive in EVA. They are more useful for internal use, but they are still public, so and they are all documented in the public help. So I will go through the other endpoints, the other categories, variants, segments, and genes, explaining some examples. ID can be a single ID or a list of IDs. If we are querying by several IDs, we have to separate them by commas. And then the resource depends on the category. Each category will have different resource available, and those are some examples: variants, info, all, summary. The filters are URL variables, so all they have to be prefixed by a question mark character, and if there are several filters or options, they have to separate it by an ampersand character. There are some filters that are common for most of the endpoints, like studies. That is a study or list of a study where you want to redirect your query. The study ID to be used is the project IDs that we use in EVA to look for the ID for a particular study. You can go to the study browser that Gary showed it before, and every study is showing their ID there. Also, for species, those are the available species that we store in EVA. They are all composed by a taxonomy code and an assembly code. Like if EVA is an archive of variants, there are many endpoints that return variants. So there are some filters common to all of them. We can filter based on annotations because all variants have been annotated by BEP. So we can filter by consequent types, or the BEP version is for annotated those variants. We can filter by minor allele frequency or polyphen or cis scores, and we can exclude or include some of the fields returning in the variant model. We will see the variant model later. And there are other options that can be used in the filters part, like URL variables. Some examples are limit and skip that can be very useful for paginating when you have a big list of results for a given query. This is the typical output of any call to our web services. The important part here is the response. That is an object that contains the number of returned results, the number of total results, because maybe not all the results for that query in the database have been returned if we are paginating using the parameters that we saw before. And the result object, that is a list that can contain strings, for example, contig names, booleans, like if a variant exists or not in the archive, or JSON objects, like the one representing a variant or an EVA study. Let's see some examples here or from our API. The variants category has 2 main endpoints. The first one is exist, that will return a Boolean telling if a particular variant is in our archive or not. We expect here the RSID, so every variant is identified in our archive by an RSID. And we can filter by species. This is mandatory actually, because each species is stored in a different database. So to redirect the query to the correct database, we need this parameter, but also you can filter by studies. So if you want to check if a variant is in a particular study, the studies filter can be used here as well. The info endpoint returns this model that we can see here for a particular variant if the variant is in the archive. So we can see that the variant coordinates are returned, also the alleles, a list of IDs containing the RSID, but also SSIDs and any other ID provided by the submitter. Typically, we will see an genomic HBS and a list of source entries. Each source entry is the information that we have for a variant for a particular study. So if a variant is in 5 different studies, we will get 5 different source entries, like in this example. This is a big object containing all the information provided by the submitter for each variant, like the frequencies or the genotypes. There is another object containing the annotation, consequent types, all the output provided by BEP, xreps, and there are other fields containing the type of and the length of the variant. The segments category is used for querying the variants in a given chromosomic region. So we have a first endpoint that is the segments ones that will return the list of contig and chromosome names that contains variants in a particular species, in a particular database in our archive. And we have another endpoint, the variants one, where the query must provide a variant or a region or list of region in that format, chromosome and start and stop positions, and all the variants that are contained in that region will be returned at this endpoint. The variants will follow the model that we just showed, and we can use the same filters that for any other variants endpoint, like the ones for annotations, the minor frequency, studies, species. In the genes category, we have also a variance endpoint that will return all the variants containing in a given gene. Again, the model return will be the same, and we can use all the filters specific to variants. All the other endpoints in our API are described in the help that is in our web. So I'm just going to go through some examples to see our API working. So imagine that we have run an experiment, and we get an BCF file containing some variants. We send this BCF to an annotator like BEP or any other, and we can see that some of those variants, this is the content of an example BCF, and we can see that some of these variants are returned with an RSID in the third column, like this one. So let's imagine that we want to look for those variants to see if they are on EVA or not. So let's extract first all the RSIDs from the BCF file. Now I'm removing the header lines from the BCF file, and I get in the third column, that is the one containing the RS. You can see that there are just some RS, and there are many other variants with no RSID. So we are getting just the RSIDs. Now we are storing the RSIDs in a temp file. We have found 3 IDs in this example BCF file. And now we want to query each variant in our archive. So we are going to call the exist endpoint in the variants category to see if each of those variants are in our archive or not. This command here, we will go through all the for each RS in the temp file that we just created. We are going to execute the call command that we call to this URL, where RS ID will be replaced by the one that for each RS in the input file. So we are doing 3 calls here, and we are going to store the result in a file named rs.json with the RS ID for each variant. Now it's calling the endpoint for the first and the second and the third variant. So we just did the 3 calls, and we can see now that there are a JSON file for each of those variants. If we see the content of the file, it's a JSON document. We can use this Python tool to print the JSON in a pretty way. And now we can see that in the response of the web service, there is a list of results just containing a result that is 2. So that variant is in our archive. We can check if a variant is in a particular study, adding the studies filter to .call. And there are many other filters. So like I'm a programmer on EVA, I know all the endpoints in our API, but for an external user, it may be difficult to find those endpoints. So for that, we have deployed in EVA a Swagger documentation in our web. If you click on the API tab, there is some API here, and there is a link to the Swagger documentation. Every single endpoint on EVA is listed here. We can see the categories that we were talking about before, like variants or studies, segments. If we click in a category, we'll see all the endpoints in that category, and we can even try them. This is the same endpoint that I just called using call search for searching a variant in the archive. Actually, is this one exists. So we can provide some parameters here, like the RSID and the species. Well, species is here because it's required. And if we click in try out, we will get the Swagger will execute the REST call, and we will see the result here. We can add a studies filter here. This is the ID for the 1000 genome study. So we can see that this variant is in the 1000 genome study. But if we use a different study ID, this is Genome of Netherlands ID, we can see that this variant is not in that study. Now I would like to show a Python example for doing a more complicated request. So we have a different test case here. Imagine that you are doing sequencing in your lab, and you are capturing some samples, some exons, or you are doing a targeted resequencing. So usually you will have a bed file provided by the target kit, the capture kit, containing all the regions that are going to be captured by your kit in the lab, and so the regions that are going to be sequenced. So in this file, each line represents a region in that capture. So we want to check here if those regions in EVA contain variants or not. So we want to stack all the variants that are on EVA for a particular study in those regions to compare with the variants that we have obtained in our sequencing experiment. I have written a Python script, a simple Python script that contains some auxiliary functions. The first one is just used for reading the command line arguments. The second one will read the bed file and will parse each region, transforming them to the format required by our API. The third function here will build the web service URL for a given region and a given study. This latest function will print its variant in the results obtained from the web services call, and this is the main method of this script. We will read the command line arguments, the bed file that are a bed file and a particular study that we want to query, and then for each region in the bed file, we will build the URL for getting all the variants in that region. And then we are using this Python library request that will execute the HTTP call to that URL that we just built, and will transform the output to JSON format that we can parse, so we can parse it. Then we will parse the response, and if there are returned results, if the number of results is greater than 0, we will print the variance contained in that result. We're going to test this script now. So it requires 2 arguments, the bed file and a study ID. We're going to use the 1000 genomes third phase, and we will see if those regions contain variants for that particular study in EVA. Now we are querying the first region, and those are the variants that have been found in that region in that study in our archive. Now we are querying the second, and we are querying the third region now. And just printing the coordinates of the variant and the main RSID. If we call again for a different study, we will get different results. This particular study doesn't include any variant for that region, and this one doesn't have any variant either. I think that I am using the incorrect study ID. So I go into the study browser to search for a given study. For example, Genome of Netherlands. We can see here the project ID, and I'm going to query now that project. Now we are getting some variants. Like we can see that this study has variants in some of the regions, but not in all of them. So thank you. Those were just 2 examples of the use of our API. You can go to the help results to the help resources to see many other use cases for our API. And now Gary is going to continue the presentation.
Okay, so now we've looked at some use cases of the EVA API, and Pablo has provided a Python script that we can find online of exactly how to do what was Pablo was doing there, which was searching certain studies for certain variants within certain regions. We have documentation on our website that provides help for the EVA API. We have a specific website which you can reach via www.ebi.ac.uk/EVA. And within the website, we have a dedicated page for the API. We also have our help page, and within the help page, we have dedicated frequently asked questions for the EVA API. Secondly, we have the EVA Web Services GitHub repo. You can reach it via this URL here. There is some help documentation here and more explanation of the endpoints that we have. Also, you can start a ticket there if you have any requests for new features to the EVA API, or if you have any general questions. We can always be reached at the help desk email, which is eva/helpdesk@ebi.ac.uk. So finally, we'll summarize the EVA API webinar. And today we've given an introduction to the EVA. We've given a detailed description of the API structure and endpoints. Pablo's given 3 examples, or the first one was a command line example, which took us through some of the endpoints that we have using curl. He showed the Swagger documentation, which is automatically generated for our EVA API, and then we finalized with a real-world example using a Python script. Importantly, that Python script and all the materials we presented today will be available online. So just remains for me to thank the rest of the people we work with here at EMBL-EBI. So our team is led by Thomas Keane. We work closely with the Biosamples, ENA, Ensemble Variation and Ensemble Genomes teams. And importantly, we'd like to thank all of our funding agencies. You can also reach us via our Twitter here, and I'll stop there and thank you for your attention.